Hello, my name is Sarah Hunkpanu, and this is my my YouTube project on the bacteria Pseudomonas aeruginosa. And at the top of the presentation, you can see a picture of this bacteria. Introduction: This bacteria is rod-shaped, meaning that it looks like little rods when you look at it under the microscope. It is gram-negative, which means that it does not retain the crystal violet stain used in the gram staining method. This also means that it has two cell walls, one inner one and an outer one. This bacteria actually turns blue-green during cultures, which is how they were actually able to detect it in the first place. It is actually very resistant to antibiotics as well. It's a rod about 1.5 micrometers long and 0.5 to 1.0 micrometers wide. It is monoflagellated, which means that it only uses like one flagella on one side of it. And the next slide, you can see a picture of what that looks like. This is monopolar flagellation, and this is bipolar and petrichous. But you can see that this is a monoflagellated bacteria. Its phylogeny, I could not get a lot of history about this bacteria. However, I do know that it did originate from Pseudomonas mendocina, and that its closest relative is Pseudomonas sp.E4. It's an isolated strain of it, and it's just a part of the family Pseudomonadaceae, and its genus is Pseudomonas. That's what I could get about the phylogeny of this bacteria. Discovery is actually very interesting. In 1850, Cedillo was the one that found the discoloration of the surgical wounds in patients, and in 1860, Force was the one that actually extracted it. In 1862, Lucky associated with rod-shaped organism, and that's how they knew that it was a rod-shaped bacteria. And in 1882, it was isolated in pure culture by Carl Guessard. And he also published an article titled "On the Blue and Green Coloration of Bandages," which actually reported the growth of the organisms from wounds of patients with bluish and greenish pus. And it was also studied by Freeman in 1916, which is very interesting. So this is the history of the bacteria. As you can see in the picture on the right here, you can see the bluish greenish discoloration under the person's fingernails, which shows that this indeed is the Pseudomonas aeruginosa. It can be isolated from very many sources. It's number 5th one that can be isolated actually, and it is actually one of the number 1 causes leading to nosocomial diseases, which means that these are diseases found in hospitals. It is the second leading cases actually in nosocomial pneumonia, 3rd for urinary tract infections, 4th for surgical site injuries, and 7th for bloodstream infections. So basically, it's one of the leading causes of nosocomial diseases that are found in the hospital. Ecological role. The flora. Actually, though this bacteria is very dangerous and resistant to antibiotics, is actually part of our flora, human flora. And on the skin, 0 to 2% is on the skin, 0 to 3.3% is actually in our nasal cavity, 0 to 6.6% is in our throat, and 2.6 to 24% is in our fecal matter, which is, I think, is pretty cool but once this gets inside your skin, it's very dangerous to you. It's a very ubiquitous bacteria, meaning that it can live in many places. It's very versatile. It does not need a lot to grow actually. It can grow in distilled water, and it can use in the clinical studies, it can use acetate as a source of carbon and ammonium sulfate as a source of nitrogen. Its temperature its optimum temperature to grow in is 37 degrees, but it can grow at actually 42 degrees Celsius. It is tolerant to a variety of physical conditions, like including temperature, concentrations of salts and dyes and antiseptics. So it's very resistant to antibiotics. In the hospitals, actually, it can grow around the water outlets, which is the way that it causes nosocomial diseases. And you'll just see the green little rust around the sink, where water comes out of, and that's one of the ways it can cause diseases. And also, new studies on plant growth shows that it's actually helpful to the plants. It actually can help them grow, but they haven't gone in depth about this yet. Metabolism. This protein actually, this bacteria has a protein called protein F, which actually it acts as a porin, which allows certain molecules and ions to come into the cell, and it lowers the permeability of the outer membrane, which is a property that is good because it does not take in harmful substances. So it's very resistant. That's why it's very resistant to antibiotics. It has a pili and flagella, which help it move for chemotaxis. They serve almost the same purpose. The pili is just long polar filaments made up of hypopolymers from the protein pilin, and the flagella is just it's like a tail kind of that just helps it move, and they kind of serve the same purpose in the cell. Then their motility in this in Pseudomonas aeruginosa is controlled by ARPAN, especially during initial attachment to the human host and under low nutrient conditions. For iron, when it invades the host, actually, it's starved for iron, and it synthesizes Pseudomonas aeruginosa synthesizes two siderophores, piocalin and pyoverdin. And then it secretes the siderophores to the exterior of the cell, where they bind to iron and bring iron back into the cell, which is another way to be a pathogen. It's a facultative aerobe, which means that it uses oxygen when it is there, but when it prefers oxygen actually. But when oxygen is not there, it also can use something else as well. So oxygen is its final electron acceptor. However, when oxygen is not there, it can use nitrate as its terminal electron acceptor. Life cycle. This is actually a biofilm life cycle. Basically, it attaches to something and then it grows. So the first stage is just attachment to whatever surface it's on. I guess in this in our body it will be our cells or where they attach, and they stay there, and then they produce a slimy extracellular polymeric substance and colonize the surface. After that, it starts developing. That's stage 2. It starts developing into a complex. It just grows bigger. Basically, it just grows, and it just it can be big within hours. And then third, it can just start clumping off. Basically, clumps of cells just start coming off just by like acidic dispersal way, and it just releases into the cell or into the body, wherever it is. And that's basically how it just its life cycle. And this is just an example of biofilms in real life. This is actually where the one I was talking about earlier with the sink, where it just kind of it just makes a living around it. It just stays there. It comes in big clumps, and you can see the blue greenish color of it around this specific picture. That's Pseudomonas aeruginosa. That's just how it becomes green. So you should check under your sink if you see any of these things. You might want to clean it out, or just call 911. So proteins involved in survival. Actually, it was hard to find information about this. I already talked about protein F, which is proteins acts as a porin, and it allows certain molecules to go in the cell and out the cell. Also, it can act as a structural protein, which helps keep the bacterial shape, the shell shape of the bacteria. And it provides the outer membrane with an exclusion limit of 500 Dalton. So it lowers the permeability of the outer membrane, and which is good because this is what helps it be resistant to many antibiotics. Another reason why is a very one of the proteins that helps it is a PA3309 and PA4352. These actually are proteins found in the Pseudomonas aeruginosa. They are stress proteins, which means that they are under stress. They are very functional. They're very good, and they are involved in survival during aerobic growth, and they arrest. They are involved in survival during aerobic growth arrest and under various other stresses. And these are the two proteins I found about Pseudomonas aeruginosa. And symbiosis. As far as symbiosis goes, Pseudomonas aeruginosa is a pathogen. It is not something you want to have. It's not an infection you want to have because it is very hard to treat and is very, very, very resistant to bacteria. It usually settles. It usually settles with immunocompromised patients, and cystic fibrosis is the most serious complication of it. It's a respiratory tract infection by Pseudomonas aeruginosa, and also cancer and burn patients also suffer infection by this organism. So if your immune system is really not it's really down or compromised, then you're most likely to get this in the hospital, of course, well, at home as well, but mostly in the hospital. This bacteria can also produce toxins, and it can cause tissue damage, but it can also interfere with your immune system defense mechanism, which is really bad for you. It is a pathogen because it can make these proteins that can toxic proteins that can just enter your cell and kill your cells at the site of colonization, and it can produce degradative enzymes that can also disrupt the cells, the cell membranes and the connective tissues. So it's not good. It's a pathogen. It's not helping anybody. Also, I did talk about how it was supposed to help certain plants, but that's still being studied, and that's about its symbiosis. And this is my works cited list for the presentation. Thank you.